Hello, my name is David Tourigny. I'm a research fellow at Columbia University funded by the Simons Foundation, and I'm here to talk to you today about several projects involving dynamic and stochastic extensions to flux balance analysis, which is as a widely used tool for computationally simulating metabolism. So metabolism is particularly amenable to mathematical representation, and that's because there's a very concrete mapping between the genome sequence of an organism and the chemical reaction network that encompasses metabolism contained within that information. And then once given a chemical reaction network, it's quite straightforward to construct a series of ODEs based on chemical reaction rate theory. And so in mathematical terms, a metabolic network with n reactions can be represented as the following ODE, where Mi represents the concentration at time t of the ith metabolite in the network. Vj is the rate of the jth reaction in that network at time t. And the stoichiometric matrix with coefficients Sij correspond to the stoichiometry of that ith metabolite with respect to the jth reaction. So in vector form, we can just write this as a simple series of ODEs equations. So the figure here taken from Clark et al. Very nicely represents a particular metabolic reaction network, simple metabolic reaction network, and its representation using the stoichiometric matrix S. And as I described on the previous slide, S then can be used to build a series of ordinary differential equations. The problem with direct solution or integration of these equations is that typically metabolic reaction networks one is interested in have a huge number of reactions and metabolites. And so not only is the dimension of these equations incredibly large, it also comes with a whole bunch of parameters that are very hard to realize experimentally. And so what researchers generally favor is certain assumptions, such as the Quasi-Steady-State Assumption, to reduce this system of equations to an algebraic system in order to be solved for the reaction flux distribution V. Now, this general approach falls under what's called constraint-based analysis, but what I'm going to talk to you about is one specific element of that, which is flux balance analysis. And flux balance analysis takes these linear constraints on V and formulates the problem, i.e. the search for a particular reaction distribution, flux distribution, as a linear programming problem, i.e. subject to these linear constraints imposed by the quasi-steady-state assumption, we're looking for a particular value of V that satisfies some biological objective such as maximization of growth rate. So although flux balance analysis has had incredible success simulating metabolism at steady state, dynamic flux balance analysis is an extension of FBA that allows the systems with dynamics to be similarly studied. And in short, dynamic flux balance analysis uses linear programming to represent intracellular metabolism, but then uses ODEs to encapsulate the extracellular dynamics that are allowed to exist in DFBA models. And in naive form, a DFBA model can be simulated as follows. One chooses a time step, delta t, and integrates the kinetic model corresponding to extracellular metabolism over that interval, after which the state of metabolism extracellularly is used to bound the constraint-based model used to represent intracellular metabolism. The linear programming problem, just like in FBA, is solved for the reaction flux distribution, which is then used to further parameterize the kinetic model for extracellular metabolism over the subsequent time interval. The problem with this naive implementation of DFBA is various numerical challenges associated with the particular choice of time step delta t, and also a series of non-uniqueness problems associated with the solution to the linear programming problem. Therefore, various researchers have proposed extensions or more exotic algorithms for simulating DFBA models. These involve algorithms that use optimal basis routines associated with the embedded linear programming problem and lexicographic optimization tools. The challenge, however, is that many scientists and researchers wanting to implement simulations of DFBA models is that they conventionally lack the expertise required for implementation of these advanced algorithms. So what we have done in collaboration with Moritz Beber and Jorge Carrasco is to provide access to these algorithms through a very nice Python-based open source interface with CobraPy, which serves as a large, widely used tool base for constraint-based modeling. So the second extension of flux balance analysis I'd like to describe is an extension of flux balance analysis that applies to the modeling of single-cell metabolism. Now, in single cells, the stochastic contributions from gene expression become particularly relevant for metabolism. And in order to account for this, we developed what we call SSA-FBA, or stochastic simulation algorithm with flux balance analysis embedded, that is somewhat reminiscent of DFBA, but now embeds linear programming problem as a representation of intracellular metabolism into simulation of stochastic dynamics. Now, given time constraints, I won't be able to go into the details of our SSA-FBA implementation and instead provide links at the end of the presentation, except to say that by exploiting and extending the advanced algorithms developed for DFBA, we were able to significantly improve our implementations of SSA-FBA simulations. I'd like to finish this presentation by thanking the collaborators on these projects and very much welcome any questions by email.